I'm Corey O'Hearn. Welcome. So I named my lab the Lab for Computational Modeling of Soft and Biological Materials. So we do exclusively computational modeling, but we collaborate with experimentalists and wet lab um, for every project that we carry out. So no wet lab in my group, but we collaborate. So my main appointment is in engineering, so mechanical engineering and material science. I'm trained as a physicist, and so I have joint appointments in physics and applied physics. With Cynthia, I'm the co-director of graduate studies, and then also I serve on the executive committee. So if the students have any questions about CB&B, I'd be happy to talk to you about classes or research or anything else. My website is there, and you can look at my papers. I keep my papers very up to date. So the first slide here, I show my current group, lots of smiling faces. And so I'd be happy to have your smiling face on this page as well. A couple of points of note, Alex Grigas in the lower right is a CBB student working on protein structure. And then some other things to point out is it's a very interdisciplinary group. So we have students from mechanical engineering like Aya in the lower middle, students from chemistry like Zhe in the lower left, and then students from physics like Yuxuan in the upper middle. So the two main projects that I'm working on now fall into two bins. One is proteins, and these are typically single protein structure or dimers, complexes of two or a few proteins. And we're interested in the structure of the protein that can be measured from X-ray scattering studies, X-ray diffraction, or NMR solution experiments. And so we have a model that's new, which basically really emphasizes the geometrical structure of amino acids, that they're actual physical objects. And it de-emphasizes more the chemical aspects, the longer range interactions like electrostatic, hydrogen bonding, or other long range. Hydrogen bonding is not long range, but other chemical interactions. And so you see here on the right that I'm really emphasizing the shape of each amino acid. And so I'm going to talk about that a specific project in a couple slides. The other main project that we're focusing on is studies of cells and tissues. And the key modeling aspect that we've come up with or rejuvenated is the fact that cells are deformable and can't be modeled as idealized shapes like spheres. And so we have really cool models of deformable particles that can move, that are modal. And so we have many possible applications for this model. One is in cancer research, another in developmental biology related to zebrafish, and another in developmental biology related to plants, plant growth. I'm going to focus on the cancer applications in this talk. So one thing that cancer researchers have are thinking about is metastasis is a real problem in cancer that hasn't been solved. You know, which tumors are going to metastasize, and it's that metastasis is linked to poor outcomes. And one way is to try to look at the shape of cells. So here we have cervix cancer, and there's imaging done where you can image the shape of the cell, but also image the nucleus and see if it's moving. And so there's some evidence that suggests that if the cells are non-spherical, if they're more elongated, then they're more mobile and they can more easily move and get around other cells. And so one possibility is just to sort of measure the metastatic potential of a tumor or a cell by investigating its shape. And so that leads into the model. Um, this is just a movie of the deformable particle model, where you can see that here I'm just compressing a tissue to make it very dense. And you can see that the cells are not spheres. We're modeling their deformable aspects. Another image, so this is a slice of breast cancer tissue. And the white sort of fat cells are actually fat, so they're adipocytes from adipose tissue. So that's like on the left. And then there's an invading tumor, which is in red or maroonish. And the tumor cells are much smaller than the adipocytes. And once they invade into the adipose tissue, they can sort of dislodge the fat cells and then continue invading. And so one thing that we're trying to do, and so now you see from the previous movie, the deformable particle cells look like adipocytes. And so the other thing that we need to model is the tumor cells. And the key feature of these tumor cells is that they're mobile. They can move around, and so we're interested in modeling their how they move around. And so here's a movie of that where we have this deformable particle. A deformable particle is made up of a bunch of, it's like a ring, and we can basically discretize the ring, and each part of that ring can move on its own. And so what we're telling here is we're telling the cell, part of the cell move in one direction, and then at one instant, and then another part of the ring can move in a random direction at some other time. And so you can see it in the movie that this cell is moving around in sort of a random way. And so what we can do with this model is study different motility strategies for tumor cells in a surrounding dense tissue. Right here, the cell is on its own. And so now here I show maybe a model that's more akin to the breast cancer slide. And so here I have the adipocytes as these white cells, fat cells, and then I have a tumor cell, and you're going to see it try to negotiate its way through this maze of fat cells. The fat cells are pinned down for now, but their shapes can change in response to forces when the tumor cell is hitting it. And so one interesting measurement that we can make is for a given time, how far can this tumor cell move in a given time? And then one other movie I want to play for tissues is we did a single cell and it's moving on its own. Then we did a single tumor cell, maybe in a background of fat cells. And here I just have a bunch of cells and they're all trying to move. This one thing you want to pay attention to is this small greenish yellow cell is one that actually breaks out of its sort of local cage and actually starts to move throughout the system. And so this could be relevant for a cancer application, but it also can be relevant for a developmental application where there's significant cell growth and the cells are moving, all the cells are growing and moving. And so in the last couple of minutes, what I can talk about is the second area where we're currently working on is protein structure. And so there are many times where proteins, the structure of proteins hasn't been determined. For example, there are many proteins associated with COVID where we haven't been able to crystallize them and we don't really know their 3D structures. And so one thing that's we can try to do is come up with computational models that try to predict those structures in the absence of experimental structures. The problem is you don't know whether those structures are right. So you'll create a model, it'll generate a low energy protein structure, but how do you know whether it's right or not in the absence of experimental structures? So what we're doing is practicing. We have a lot of experimental structures of proteins and we have a lot of computational models, and we're trying to figure out whether those computational models are accurate or not, both with and without looking at the correct answer. So on the vertical axis here is a score. It goes from 0 to 100. 0 is a bad score, 100 is a good score, and it's a way of ranking protein models. And in this case, we're looking at the answer. So you can just say, here I have a computational model. I compare it to the real answer, and I can give it a score. And so this is a recent, what's called a CASP competition. And what they do is, it's a protein folding competition. They have an experimental structure, but they don't release it to the community. And so you generate your best models and then compare, then the competition compares your answer to the real answer. And so in general,
it's 9:12.
Okay, it's 9:12. So I'll just finish up talking about this. And so what it says is that current protein folding is not doing that well. It's not consistently 100. And so there are a lot of bad answers. And so you have to filter the good predictions from the bad predictions. And so what we've come up with, a sort of crazy but interesting idea that we've come up with, is that maybe you should just focus on the core of a protein. Instead of focusing on the whole protein, maybe the core acts as a template. And if you get the core residues right, the ones that are buried and hydrophobic, then maybe the rest of the protein just follows on as a template. And so you see here, you see that on the right plot to some extent works, that we look at the deviation of the core residues from the right answer. And if the core residues deviate by a little bit, you get a really high score for the answer. And so with that, I'll finish up. I'm happy to take on any rotations that people want to do, either in the tissue and cell-based work or the protein work, and just send me an email to talk about it. So I'll pause for a minute if there are any questions.